go to the slide that I skipped before in order to let you understand. I hope someone here in this classroom would like to be a pharmacologist in the next future. So now I'm going to give you some small but quite important hint how to do PKPD study. First of all, think about this. I have to select my animal, cat. I'm going to the shelter and the shelter owner give me 12 animals. And I have to divide 12 animals in two groups, two different groups. How can I divide the animal if I want to have two randoms groups? How can I? Come on, you have 12 animals. What should you do practically? Don't be shy. What should you do? Yeah. You can use the computer and randomize the animal, but how can you randomize the animal? Or if you have not a computer, you can, write a number in the bag, we draw the number. How can you do it? Before you have to give a number to each animal. So take cat number 1, take one cat and give number 1. Take second cat, give number 2. But be attentive. You cannot just take the first 6 animals by chance and do one group, and the 6 other animals and do another group. Why? If you do this, what can you generate? Because it looks like a randomization, because you are taking the animal by chance, right? But it is not. Why? In this case, you are generating first group, that is the weaker animal, and second group, that are the strongest animal, because cats doesn't like to be touched. So they're going to escape. And who are you going to take first? The weakest. And another week, and so on. So this is not randomization. Randomization is give to each animal one number, 1, 2, 3, 4, 12, and then we draw number 3. Doesn't matter. Until you didn't get number 3, you cannot go ahead. Number 5, and so on. This is randomization, so remember. And then, when you have 2 groups, you can run parallel study or crossover study. Have you heard about this name, parallel study or crossover study? Do you know what it means? This is a parallel study. It means that your 2 groups has been divided in 6 animals administered by EV, and you do one experiment, and later you have other 6 animals that are divided in the intramuscular or extravascular administration. But you can also do, that is much better, crossover study. Crossover study can be done So look at here. We have 6 animals, so less than before, and we use 3 animals by EV and 3 animals by EM. At the same time, then after washout period, that later I'm going to tell you how to calculate, the animal that got the intramuscular administration get into the intravenous group and get the intravenous administration. And the animal that got the intravenous administration get the intramuscular administration. What means? It means that at the end, 6 animals have both intramuscular and intravenous administration, right? Why this is much better than the parallel study? You have an idea? Well, think about this, the first one, the parallel study. Let's assume that during the first experiment, we have full moon. Full moon can affect maybe negatively the animal. So I have a big bias due to the full moon. But the full moon is not present in the second time when I do the second group of experiments. So I can, through my data, think that the effect of the moon is not due to the moon, but is due to the intravenous administration. So in this case, we have to be, or we are expected when you use a parallel study to have a big bias than a crossover study. Because if the same situation is done in the crossover study, watch here, the full moon will affect these 3 animals. So it's not a big matter because I will have the affection of the full moon in the 3 animal intravenous and in 3 animal intramuscular. So they will be compensated, right? And how much time I have to wait until I can reuse the same animal? Because in the parallel study, I don't need it. Where is the parallel study? In the parallel study, I don't need it because they are different animals. So I can do, for example, this experiment today and the other experiment after 2 days. Doesn't matter because the animal will get the drug for just one single time. But in here, I have to be really attentive because these 3 animals get at the beginning intravenous and after sometimes they get the intramuscular. So I need that a time will be spent in order to allow the drug that has been given the first time to be completely eliminated before the second administration. How much is this time? Why 3 weeks? Well, 3 weeks, I don't know why you said 3 weeks. I can give you some example that can be 1 day, or some other example that can be 3 months. And it's not depending on the drug effect, but it's more depending on the drug plasma concentration. So I know that you are a beginner, so you are not accustomed to this. But there is a pharmacokinetic parameter that is named half-life. Do you heard about this? Half-life. What is the half-life? Do you remember the definition of half-life? Is the time that reduce of 50% what? The what? The plasma concentration of the drug. Okay? So, watch here. So you are going to remind this. That is quite important. At the beginning, I have plasma concentration 100%, right? After one half-life, I have 50%. The second half-life, I have 25. Then 12.5, then 6.75. Now 3.30 something, fifth half-life, and then 1.76 half-life, and seventh half-life I have less than 1%, 0.8 something, seventh half-life. And then I could go ahead, but I'm not good in math, so it's better to stop here. So what we consider as, first of all, think, when the drug concentration is going to be 0, when? Theoretically. Come on guys, theoretically. When is going to be 0? 50%, then 50%, 50%, 50%, when is 0? Never, theoretically, never. Because you're reducing 50%, 50%, 50%, and so on. But practically, we can consider that really small amount of drug is still left in the plasma concentration of the animal, because less than 1%. So it is expectable that this drug concentration cannot affect the second injection of the drug when I run a crossover study. So, getting back to the 3 weeks that this lady said before, is not the correct answer, but the correct answer is from 7 to 10 times the half-life. If the half-life is 1 week, at least 7 weeks, from 7 to 10 weeks. If the half-life is 2 hours, from 14 to 20 hours. Okay? Just because the plasma concentration are lower than 1%. So keep this in mind, because the explanation is quite easy to understand. So, and now let's go to this slide. He won't attend as well. But he doesn't know that he's a classroom of pharmacologists. So we can do an experiment. It's better if he doesn't enter here. So, look this. This is a parallel study design, just to show you. So we have 4, 4, 4, and 4. 4 groups of 4 animals. We administered the dose to this animal, dose number 1, and we have a response. If we administered the second dose, or twice the previous dose, we have an increase in the response, and you see the significance of the result. The third and higher dose still significant, and you can see here that the fourth dose is not statistically significant with the third. So it means that here I have achieved the maximum effect. But in here, look, I can run a crossover study. With a crossover study, each animal can get all the doses, all the 4 doses. So each animal is control of itself. And it's much better because with the crossover study, I can design also these curves that describe the Emax 50, E50 and EB50 here. So it's really important when you do this study, if you approach a crossover study rather than a parallel study. Welcome, Professor Arnold. Welcome to this lecture. So, let's go back here. Now, which is the difference between the dose titration that I told you before at the beginning of this lecture and the PK/PD study? Both can determine the maximum effect. It's not a big issue because with the titration we give the dose, increasing the dose of the drug, and then when you see that the effect is not changing anymore, you say, okay, this is the Emax. But the same can be done with the PK/PD. But what is important is that with the dose titration, we are dealing with the dose. But with the PK/PD, you are dealing with the concentration. And this is exactly what you need, because the dose cannot be, cannot be switched between different species, but the concentration can. So this is a big issue. And you have to figure out the potency of the drug according to the concentration, much better than the dose. And then only PK/PD can give you the selectivity of the drug. And I'm not sure if you already taught them the receptor selectivity, potency, and so on, or if they are going to learn this later in the time. But what is important also is this one that I told you at the beginning. This is still the study that I showed you with the cat. So we did only one single experiment with one single dose, and thanks to the PK/PD integration, we can do the modeling. What is the modeling? The modeling is I put different doses in the computer, and the computer can give me different effect because of the different dose that I have inserted in the computer. So I don't need to run any more experiment. I have just to insert, 0.1, 0.2, 0.3, and you can see here that if I increase the dose of the drug, the effect is going to be increased. Indeed, 0.1 has a pain score 100%, so it means that the animal is in pain, is fully in pain, the drug is not effective. But if I give 1 milligram per kilogram, you can see that the drug is effective up to, and even more, 25%, because the pain reduction is really, really important. And you can see here. And the same, what about if you want to split the dose? We gave 5 milligram per kilogram once a day. And the 5 milligram per kilogram is this one, so is the purple one. And you can see here that the mean effect was 32%. So it means that in 24 hours, if we administer the drug 5 milligram per kilogram once a day, we have a pain reduction of 32%. What about if I administer 2.5 milligram but twice a day after 12 hours. And you can see with the blue line, the mean effect in 24 hours is 52%. So exactly the same dose, but split and administered 12 hours of time. And what about if I divided the dose in 12 different doses, but the total amount is still 5 milligrams per kilogram? Look here, the green one. Look how, of course, the effect is reduced. And the total effect in 24 hours is a pain reduction of 96%. Now you can wonder, how is it possible to take a cat and administer it every 2 hours per day the drug? Well, it's impossible, of course, because the drug is going to kill you, very aggressive. But in this case, the pharmaceuticals can give us help. They can produce some sustained release formulation that you can give just once a day the drug to the cat, and the drug is going to be released smoothly and constantly from the gastrointestinal system. So simulating this green line. So that's the reason why the PK/PD are important, because also from this, it's quite well understandable that you need a sustained release formulation to improve the effect of the drug. So, let's go now to, we saw PK/PD for the non-steroidal anti-inflammatory drug or for the painkiller drug, and let's check now the PK/PD for the antibiotic. PK is PK, so there is no difference in between non-steroidal anti-inflammatory drug and antibiotic. Both have a half-life, volume distribution, clearance, and so on. But what about PD? In the effect that we have considered for the non-steroidal anti-inflammatory drug or painkiller drug, we got some final effect that were the total pain, that were, remember, the numbness, it were the swelling of the paw, the heating of the paw, so different kind of final surrogate effect. In the case of the antibiotic, which kind of PD effect are you going to consider? Which is the effect, final effect of antibiotic or antibacterial drug? Why you take an antibacterial drug, or when you take an antibacterial drug? When you have an infection, bacterial infection. So, which is the mechanism? I don't want to enter in detail, which is the 50S, 30S, blah, blah, blah. Which is the final effect of antibiotic? To inhibit or kill the microorganism, the bacteria, right? So, which are the final effect, I mean, what we can use? The MIC, minimal inhibitory concentration. I guess you have done this in microbiology, minimal inhibitory concentration. You remember this? No? Yes? Someone yes? Someone no? Minimal inhibitory concentration, or MBC, minimal bactericidal concentration. But it's also true, watch here, that when I use an antibiotic, I need to be really attentive because I want that the drug could be effective, but I don't want to increase too much the concentration because it can be toxic. We are dealing with animals. Most of them can be infected because they are, bred in intensive breeding system. So they are food-producing animal, and we don't want the residue in their meats, in their goods. And then what we want also is the lack of bacterial resistance. That is a topic that is really fancy nowadays. You know from the media that nowadays there is a big issue with the use of antibiotic veterinary medicine, because people think that the wrong use of these drugs can have been caused the bacterial resistance. And if we have a lot of bacteria that are resistant, the drugs are not more effective. So, how can we use the antibiotic? It's not so easy to use antibiotics like to use a painkiller drug. It's much more difficult, and what we call judicious and rational use of antibiotics. Not all the bacteria can be destroyed, can be inhibited by a single class of antibiotics. So first of all, for the selection of antibiotics, we have to run a microbiology study in order to understand which is the bacteria and which kind of bacteria has infected the animal's body, what kind of susceptibility has this bacteria for all the class of antibiotic. When we found that this bacteria is susceptible to one single molecule of antibiotic, we select that antibiotic because it's the more important for the bacteria inhibition or bacteria destruction. But unfortunately, often we can have some problem with this approach, because from time to time we have to deal with animal species that has never been tested. Nowadays, your professor of pharmacology is dealing with crocodiles. No one knows what's happened when you inject one drug in crocodiles, which is the bioavailability, which are the toxic effect, which is the real therapeutic effect. So in veterinary medicine, the off-label use of the drug is something that can be used often. And you have to keep in mind that this can generate, if not well done, antibiotic resistance. And here you can see different kind of causes that can generate bacterial resistance or can generate the therapeutic failure. So, as we said before, for the non-steroidal anti-inflammatory drug, there is a black box between microbiology and the therapeutic effect of the drug. And we have to open this black box, and we have to understand what there is in this black box. This black box, we can find PKPD, like before. So I don't want to get back to the 4 processes of the pharmacokinetic that we already summarized before, ADMI, but just to let you know the most important parameters, because you are beginners in pharmacology. This is the Cmax, so maximum plasma concentration of the drug, we already said. This is the Tmax, time at which the Cmax is achieved, and more important, because this is pretty important for the antibiotic, this is the area under the curve. It's pretty easy to understand what Cmax and Tmax are, but which kind of information brings you the IUC, area under the curve? Is the extent of the drug within the time. So all this area is the extent of the drug in this period of time. And this is pretty important for the antibiotic. I'm going to show you why. I'm going a little bit faster because otherwise we have no time. Here, for the PKPD indexes, as I told you before, we can use the minimum inhibitory concentration, MIC. It is pretty important every time that you want to assess if one bacteria is susceptible to some antibiotic. And the minimum bactericidal concentration. Remember that minimum inhibitory concentration means that the bacteria growth is flat, so the bacteria cannot be multiplied. But the minimal bactericidal concentration means that you are going to kill them. How many of them? Someone could say, everyone. No, that is eradication. Minimal bactericidal concentration means that you're going to kill 99.9% of them. So still 0.1% is in your sample, right? How can you calculate the MIC? If you remember, no, you don't remember. By the way, it's something that you can do in the lab, not you, but there are some labs that can do it quite easily. So the bacteria are diluted in these tubes. You can see these are bacteria and incubated for 24 hours. In this tube, you can see there are different concentrations of the drug, from the lowest concentration, 0.5, up to a multiple of 32, even 64. You know, double, so 0.5, 1, 2, 4. Each concentration is double than the former one, the previous one. After 24-hour incubation, what you're going to see? If you see that your sample is not clear, like in this case where you can find such kind of particles inside, it means that the bacteria are still there, they are multiplying. If it's clear, it means that the bacteria does not grow. So it means that this is the MIC of the compound. It means that at 8 microgram per ml, the bacteria is inhibited. They cannot grow. And this is the plasma concentration that you have to reach in your animal in order to kill this bacteria, not kill, to inhibit this bacteria, right? This is for the MIC. For the NBC, you are going to take these tubes where you didn't get the bacteria grow, incubate again for 24 hours and check what's happening. And then you can calculate the NBC, minimal bactericidal concentration. In that case, you have killed the bacteria, not inhibited, but killed. This is what we call the lab test. There are also other kind of easy tests that can be quantitative or semi-quantitative tests. For example, look about this. This is the Kirby-Bauer test. Each of these small discs has an antibiotic inside. It can be different concentration, can be different antibiotic. And in this Petri dish, you have bacteria spread everywhere. After incubation, you can see here a sort of fade rounded shape. It means that here bacteria didn't grow up. So according to how large is this shape, this shadow, you can calculate how many bacteria has been killed or has not been grown up. But this is not quantitative, it's a sort of qualitative analysis. But really easy to do. I mean, you have just to spread the bacteria there, put these small rounded sort of tablets with antibiotic inside, and let rest for 24 hours and 30 seconds. Morning later you have the results. Of course, if you want to check, this should be susceptible, because you see that no bacteria has been grown close to the antibiotic, to the paper disc, and this will be resistant, because this drug looks to have no effect on the bacterial growth. Then we have other kind of advanced diffusion test, in which this paper has, you can read here, a different concentration of antibiotic. Here, very, very small, and increasing up to the top of the label. Where is the MIC? The MIC is around here, 0.64, 0.84, 94, I cannot read very well here, because it's exactly here where you have grown of the bacteria and not grown of bacteria. And this is the microdilution test, what I told you before. So in this plate, we have different concentration of the drug with the bacteria. Where is the MIC here? I told you before in the slides, is where we see the transparent solution. It means that here we have not bacteria grown. So the MIC will be here. And here, and so on. Now, let's do a test. This is exactly the same dish that I showed you in the former slides, in this one. And this is the drug concentration contained in each well. Where is the MIC here? Consider that this black spot means foggy fluid. So, which is the MIC in this A row? 0.03, 0.5, 8. Come on, someone. Don't be shy. Yeah? Exactly. Still. What about the B row? 4? Look well. How is it possible that you have no grown here, and at higher concentration you have grown back? So you have to repeat. This happened. This happened. What about the C? Exactly. What about the D? What about the E? Lower than. What about the F? Don't use this drug because not effective. Not effective at all. No, they are still alive. No, if you want to reach more than 64 micrograms per ml in the animal's body, it means that you have to administer a huge amount of drug, so don't use it. You need some more drug in which the bacteria is more susceptible. And what about G? Something strange, it's not exactly like this. So when something this, some interference, it's better to be repeated. Last one. Watch well the last one. Come on. If the last 2 points are controls, which is the MIC? Exactly, 0.06. Good. So, what we said so far? We said that this is exactly what happened with the antibiotic. We have MIC and MEC, of course, MIC is a threshold lower the MBC, right? So the plasma concentration of the antibiotic should be higher in MBC if you want to kill 99.9% of the bacteria. So if I'm in the bottom part of this graph, I want to say in here, what's happened to the number of bacteria? Are they increasing, decreasing, steady state? Why steady state? I'm above the MBC, so concentration higher than the minimal bactericidal concentration. So if I kill 99.9% of the bacteria, if I'm at the MBC, if I'm even higher, it means that no, they should be dropping down, right? And what about if I'm in between MBC and MIC? Steady? Why steady? But, you know, I'm over the MIC. Because I'm over the MIC but below the MBC. So I'm not killing 99.9%, but I'm killing 50%, 30%. So they are dropping down, but with a slope that is not so, you know, is shallower than this. Because I'm still killing some of them. What about if I lower the MIC? Yeah, he said, right, theoretically. Because he said, I'm lower the MIC, so if I'm at MIC and they're steady, lower than MIC, they start to rise again. For some antibiotics, that's true, but in experimental condition and also in vivo condition, your animal, you can have this. Why? Why? This is what we call post-antibiotic effect. And why you have? When I explain to my students this effect, I do this example. Think the bacteria like soldiers. They are going into the war. They are bombed, they are killed. Most of them, 99.9% are killed, and they are still going to be killed, and so on. When they are here and the bombing is quitting, most of them are still injured, are still weak, super weak, because they are starving. They have been, as I told you, bombed and shot. And then, even if the drug is lower than the MIC, still have an effect on some of them that are very, very weak due to the high concentration of the drug before. So, you have a reduction. But, if some time is spent, they start to regrow, to rise again. So, remember that not all the antibiotics, most of the antibiotics can also have the post-antibiotic effect. Okay. But now you can wonder, what is important for an antibiotic? Is it important to be high with the concentration, or to be really close to the MIC to not induce side effect? So, how should I manage the situation? Increase very, very much the concentration, Cmax for example, or maybe to extend the time in which the drug concentration is over the MIC, but keep this concentration really close to the MIC. How should I behave? And for this, we have three I should say two, but for your convenience, it's easier to be explained. I tell you three kind of different antibiotics. Antibiotics that are concentration-dependent antibiotic. It means that higher is the concentration of the drug, so higher is the Cmax, more they're effective. So therapeutically speaking, I have to give really high concentration of the drug and we try to increase the Cmax. They are much more effective. There are other kind of antibiotics that are concentration and time dependent. So it means I have to increase the Cmax, but I also have to increase the extent of the drug during the time. So in this case, IUC is the parameter that is really important, area under the curve. And the third one are the antibiotics that are time dependent, in which it's really important to understand that I have to increase the plasma concentration just a little bit below the minimal inhibitory concentration and keep this concentration steady as long as possible. So do not increase a lot the plasma concentration like the first and the second category of antibiotics. Just for your explanation. This is what is important for the concentration-dependent drug. And of course, the PK parameter that is important is Cmax over MIC. Cmax is a PK parameter, MIC is PD parameter. So we call it this PK-PD surrogate. Then, concentration and time dependent. PK parameter is the IUC in 24 hours, PD parameter is MIC. Keep in mind that the MIC is always the same because it cannot change if you are dealing with the same bacteria. And then the time above the MIC over time above the MIC are the third category. So first one, Cmax is important. Second one, IUC is important. Third one, D. You can see here, from here to here, where the concentration is above the MIC is the important parameter. Now, from this graph that I'm going to explain you have to tell me if this antibiotic is concentration dependent, time dependent, or concentration and time dependent. Let me explain before the graph. This is an experiment that we do in the lab. So here you have the amount of bacteria. At the beginning are always the same. So this is 1 million bacteria because it's log base 10 colon forming unit per ml. Each ml of this solution contains 1 million of bacteria. So if I don't put any drug during the time after the incubation, look, the bacteria are going to have a party, to have sex, to replicate together, and so on. And they increase in number. If I go 1/4 of the MIC, they replicate, but a little bit less. They grow, but a little bit less. If I put the MIC concentration, there, of course, this is a lab stuff, so theoretically it should be straight. It's not exactly straight, but as you can see, it's quite steady, the amount of bacteria. And then 4, 16, and 64 MIC, they have the same decrease in bacteria number. So, is this antibiotic time dependent or concentration dependent? Time dependent or concentration dependent? Concentration? High concentration. Concentration, look, 4, 16, and 64 different concentration, but same profile. So it is not connected with the concentration. It's connected with time, because 2 hours, they are still there, 4 hours a little bit less, 6 hours still less. So I'm sure that if we go ahead with the time, we will have decreased more. So this is time dependent antibiotic. And you can clearly understand from here. This same experiment, but as you can see from the graph, is important the time, but it's important also the concentration, because 64 generate the destruction, the killing of bacteria, much more than 16 mg, much more than 4 mg, and so on. So this is time dependent, this is concentration and time dependent. But now let's check if you are smart enough to understand if the drug, in this case is ceftazidime, still same amount of bacteria, the beginning here, and then here what I do, I'm going to increase the IUC over the MIC, so the PK/PD parameter that stands for concentration and time. Here I'm going to increase the PK/PD parameter that stands for concentration-dependent antibiotic, and here I'm going to increase the time above the MIC for the same experiment for the same drug. Is this drug concentration and time dependent, concentration dependent, or time dependent? It's super easy. If you trust me, well, when you know this, the solution is super easy, even more. But also now, what can you see? Which is the difference between this graph? The color is the same spot. How the spots are located? Can you see any difference? Look here. It means, MIC always the same. What should I do if I want to increase the IUC? I have to increase the dose of the drug, right? The same here. MIC always the same, it's a fixed parameter. If I want to increase the Cmax, I have to increase the dose of the drug, and here as well, I have to increase the dose of the drug if I want to increase the time above the MIC. So, in all 3 cases, I have to increase, if I want to move from the left to the right, the dose of the drug. But, which is the effect on the bacteria? Look here. The spots are spread everywhere. Of course, if I increase the effect of the drug, it means that I have an improvement, so a decrease of the bacteria. Here as well, but still the spots are spread. Look at the correlation that we have here. Can you see the correlation? It's a straight line. So, it means that this drug is a time-dependent antibiotic. When you use ceftazidime, you have to use, keeping in mind that it's much better to keep constant concentration for a little bit higher than MIC for longer time, rather than drastically increase the dose and increase the Cmax. Now, I'm sure that you are going to fix this. Is this drug concentration and time dependent, concentration dependent, or time above the MIC, so time dependent? Same experiment. Come on. First, second, or third? First, right. I told you, it's easy. Because even here, you can see that you have a really straight correlation. And the best fitting is from the time concentration over the MIC parameter, rather than time above the MIC. We are almost there, last two slides. But these are really difficult, so I will try to be, okay, we can skip this and I can tell you this. Now, last slide. This is a drug that has been shown to be time above the MIC, so time dependent. But now, you are the clinician, the future clinician. How long the plasma concentration of this drug have to stay over the MIC within 24 hours? For all 24 hours or less? Maybe for 12 hours, or maybe for 10 hours, or maybe for 5 hours is enough? This experiment is going to tell us how much. Look, this is 24-hour time expressed as a percentage. So 100% is 24 hours. 0 is 0 time. And in here, we have the mortality of the bacteria after administration of 2 antibiotics. But just check, for example, the penicillin, the orange spot. Don't consider the cephalosporin. So what it means, that if my bacteria are left for 50%, 50-60% of 24 hours, that correspond, how much is 60% of 24 hours, should be higher than 14 hours around, no? We have a reduction in mortality of the animal infected with this Salmonella pneumonia, up to all of them survive. And then from 60% to 100%, similar situation. What it means? It means that if I have saved all my animal after 14 hours of concentration above the MIC, and I have no more improvement, why should I keep the concentration still there, still high? I can just have side effect. So, in this case, I can just keep the concentration above the MIC for 60% of the 24 hours. Then, doesn't matter, because I already achieved the maximum effect after 14 hours of treatment. Then, it's not anymore my matter, because the antibiotic has achieved its maximum. So, remember that most of the drug don't need to have a time above the MIC for all the 24 hours. Usually it's from 60 to 70% of the 24 hours that correspond to 14, 16 hours. And the same can be done, and you can see here. Here we are at the steady state, so all the animal has been saved. And the same can be done also for this. That is the parameter IUC-MIC. How much big should be IUC-MIC? Well, usually in here should be around 100%, 100. So it means that your IUC, that correspond to the extent of the drug divided the MIC, should have a value of around 100. If it's 100, doesn't matter to go to 1,000. If you go to 1,000, means that you have to administer 10 times more drug than for reaching 100. Because the IUC is strictly correlated with dose. IUC is dose proportional. If you double the dose, the IUC is doubled. If you give 10 times the dose, the IUC is 10 times higher. So, this, for example, is the KPD threshold that we consider for the fluoroquinolones, like enrofloxacin, ciprofloxacin, marvofloxacin, fluorofloxacin. We consider that the drugs are effective when the IUC-MIC is around 100. And this can be really important for example, new animal species in which you have no idea how much is the dose that you can administer. So you administer the dose. If the IUC-MIC is lower than expected, you do the calculation and you calculate immediately the right dose that brings you to IUC-MIC ratio around 100. And then you can use this antibiotic in different animal species. I know that is a little bit complicated, especially because you are at the beginning and you have not approached still these antimicrobial drugs. But my hint is that once you have approached the antimicrobial drugs, please get back to this lecture and see these slides again. You will see with a different view, you will understand better. And of course, I will be present in any time. Professor Popolatev has my email and everything, and he's more expert than me, so you don't need me. You can reply to all your questions. So, if you have any question, yeah, I'm pretty finished. If they want.
We appreciate if you have any patients. The position for you is the expert for the PK and PD, so that you should have patients that I told you in the morning. Any questions? Please, don't be shy.
Okay, very good, student. You can use a microphone.
I don't quite understand the clockwise hysteresis loop. Can you please explain it again?
Clockwise?
The clockwise
is,
one.
okay. Your colleagues before got the same problem. My fault, sorry. So, oops. What you see here is the clockwise hysteresis loop, where the effect came first, then the increase of plasma concentration. How is it possible? We said that usually in the anti-clockwise hysteresis loop, before we have, and that's quite pretty normal to understand, increase in plasma concentration, and then maybe late, increase in the effect. In this case, we have increase in the effect and later in plasma concentration. So, let's assume that we have the parental drug, PD, and one metabolite, name it M. So, in the case of clockwise hysteresis loop, what's happening? When you administered the drug, PD, parental drug, the parental drug gets converted in metabolite. So, if at the beginning, at time 0, you have 100 molecules of parental drug, immediately it gets converted in metabolite, right? So maybe 50% are converted in the metabolite. So after a few seconds later, you will have 50% of the parental drug and 50%, 50 molecule of the metabolite. But what's happened if the metabolite is more effective than the parental drug? In this case, you see that the effect due to the 50 molecule of the parental drug plus the 50 molecule of the metabolites is going to increase very fast because the metabolite is much more active than the parental drug. But what you see that the concentration of the parental drug is going to drop down because it's going from 100 to 50 because it's converted in something different, something that is not anymore, no longer the drug, the parental drug that you got at the beginning. That's the reason why. Clear now? Clear for you guys?
Are you okay? Is it gonna be fine? Okay. Other questions? So, did you explain about the flip-flop phenomena for myself?
Yes.
Could you please? Yeah.
Professor Ammer asked me to explain you what we call flip-flop phenomenon. We already spoke a few hours ago about the half-life. The half-life is, now you should know, it's the time that reduce of 50% what? The plasma concentration of the drug. How this half-life is calculated? These are the drug concentration over the time, and these are experimental point, so withdrawal point for the pharmacokinetic. This is the pharmacokinetic curves, and usually from the last 3 points that are expected to be complete elimination part of the curve, I can calculate the half-life. This is pretty good if we do after the intravenous administration. Why after the intravenous administration? Because the intravenous administration has no any absorption phase, right? Because at the time 0, you inject all the drug into the bloodstream. So you have no absorption. You have just distribution in the first part of the curve and then elimination. But when you administer the drug in extravascular administration, that can be intramuscular, oral, rectal, in the eyes, or whatever you want, you can have that the same drug show like this, a longer half-life. Why? Can you try to guess why it's happening? Because in this case, in the blue line, The drug balance of the blue line can be expressed as drug in and drug out. At the beginning, so in this part, drug in is much more important than drug out because the plasma concentration starts to rise. So drug in is much more important. But also drug out takes place because as soon you start to absorb the drug has an important absorption that increases the plasma concentration, but some small part of the drug can also be already eliminated. When you arrive here, drug in is equal to drug out because you are a Cmax. Exactly in that point, drug in and drug out are exactly the same. But look here. In here, drug out is much more important than drug in. But the drug in, unfortunately, in this case, is still important and affect the elimination phase of the drug. This sovereignly happens, for example, with sustained release formulation, in which the drug is constantly released, time by time. And in this case, we call it flip-flop effect because the absorption in this phase is still important, is not negligible or absent like in the intravenous administration. Now you can wonder, how can I evaluate if I have the flip-flop phenomenon or not? Well, you have to compare the half-life calculated after intravenous administration and the half-life calculated after extravascular administration. If the one calculated after extravascular administration is much longer than the one calculated after the intravenous administration, it's probably you have a flip-flop phenomenon. So the half-life that you have in here, this half-life here, is not a sound half-life because still include the absorption that is still releasing drug. But the only reliable half-life value is the one that is calculated after intravascular administration. Of course, we can also have drugs in which this effect, so the flip-flop phenomenon, doesn't exist. So intravascular and extravascular administration get exactly the same half-life.
Thank you very much. Any questions? Maybe this already is going to be okay. And then please let me or give me the best student for your class to say something to Professor Giorgi. Dion, okay. Dion is very good student, very high responsibility.